Only half days.
Good morning, everybody. Our first talk of the morning is by Jörg Stelling. He's going to talk about analysis of self-variability.
Yeah, thanks a lot, Ruth and Jeremy, for the invitation. I'm going to look at a class of models today. We're not introduced to years apart. But the main focus is analyzing self-variability. So just to define this a little bit more precisely, we're looking at cases where we have cells that are genetically identical, and we assume that they live in the same environment. So when you now observe variability, like in this picture here, these are yeast cells, there are many different possible causes. So they may simply be intracellular stochasticity, but they may also start from different initial conditions, and they may be in different cell cycle stages, and they may have different ages that explain part of this heterogeneity. The question we're now trying to ask is, because that's biologically and also from an engineering perspective really relevant, is what are actually these causes for given examples, and how can we try to figure out what these causes of variability are. So the experimental background, just very briefly, that's from our own experimental data. That's what we do. For instance, under the microscope here, you see a cell, this green one here. We do time-lapse imaging. We follow these cells over time, evaluate things like the fluorescence intensity here over time, and at the same time, we see how this colony essentially expands. We get the lineage information here, so that's again the green cell. They give rise to daughter cells, and we get essentially this complete information. In order to be able to do this, just to give a little bit of hint, Johan said this yesterday, there's a lot of infrastructure behind this to enable it. On our side, there was a lot of effort in essentially developing software, so ranging from image segmentation and trying to establish these tracks, these lineages, to microscope control and to data integration in the laboratory information systems. So this is all open source, and that is interested just as it talk about this more. So go back to the classical experiments by Elowitz and Leibler from 2000 here. So what they did, they just took a bacterial strain, putting two genes, one essentially encoding for the red protein, another one for a green protein, imaged over time, these two fluorescences. And then it came up with this scheme here that they divided the noise, the variation over time, into an intrinsic component, which is intrinsic to this one gene of interest. And you see it essentially here. In this case, if it was all intrinsic, then these two traces wouldn't be correlated. If everything was driven by some extrinsic, and extrinsic is really with respect to one gene of interest, stochastic process, then you would see high correlation. So this gives us a handle. So we can now just plot fluorescences against each other. Everything that is extrinsic would be here on the diagonal, so the correlation. Everything intrinsic would be in this delta. So that holds only for this particular type of experiment, but in the literature there's a lot of confusion of what is meant by extrinsic, intrinsic. So we extend this picture a little bit. So here in the middle is the traditional model where we have DNA that gives rise to mRNA and transcription with degradation of mRNA, eventually protein. So this would be the intrinsic system of interest. We're looking at here, you have variability. Now, what is shown here is stochastic noise. On one hand, in these published traces, and then just three different cells in a population. And there are essentially different factors that influence this noise that you eventually will observe. So this is the measurement against the three cells. On one hand, of course, the intrinsic stochasticity of these reactions, but also essential components such as polymerases and ribosomes influence. So they can be different between different cells, and they also have their intrinsic variability here. So they essentially So there are different sources of heterogeneity the cell growth, cell cycle stage. So it's really hard to explain when you have this observation here, whether it's just due to variability here, due to measurement noise, or due to different stages of the cells that eventually, by this route here, influence what happens in this system.
Is that experimental data or simulation data?
That's just simulation. That's what you might observe here at discrete time points. It's just
That is experiment. The field C, field B, field A, all of these are simulation.
all of this is for illustration. I'll show you real data in a few minutes.
Is there any particular reason that even in your simulations, polymerases may change so much compared to ribosomes being more constant? I don't know. It's just funny. Why did you do that? Just an artist's view? Okay.
Okay, so this is much more highly regulated in biology. So
Okay, that's what I'm saying. It is more highly regulated as RNAP.
Okay, so the point here is really that there are these interconnected processes. It's hard to figure out what the origins of this observed variability between cells and mutation are. And just to give you a few examples, if you take this standard model of gene expression, so we have production of a protein, degradation of a protein, so that's a linear and dynamic system. What is shown here are stochastic simulation results. So in red here, the one trace highlighted here, and this would be the mean trace, mean of the data. And what we assume usually in the stochastic simulations is that the parameters, so the production and degradation constants here, they are identical for every set. Okay, so that's illustrated here, just one set of parameters. If we run this stochastic simulation here, we know what the result is. It's what the Poisson distribution in the steady state. But if you now assume just simply that these parameters here are distributed in a certain way between cells, measurement noise on top, the system is completely deterministic, it's just an MLE system. The observations are in distribution. It becomes more interesting when we have more nonlinear system. So this is essentially an extension here. It's Y here, and then you have a positive feedback here, so it's essentially bi-step switch. Now again, so it gets the noise, you get the variability here. The main difference now is that the behavior of an average cell, so a cell that has the average behavior here, is different from the average of the observations, and simply because it's a nonlinear system. And you see the same thing here when you vary parameters again. The last point with these illustrations here is these three scenarios, they're all deterministic. Again, the parameters are distributed. The total variation in the parameters is the same in all three cases, but the covariance of the parameters is different. Here it's just one dimension. Here it's essentially a normal distribution around the center, and here they're highly correlated. And you see that the dynamics and the individual trajectories can differ a lot. It's not so the correlations are important. When we assume that different cells, for instance here, you can imagine two genes being expressed, and they're correlated via, for instance, how many ribosomes there are. Conversely, this may give us a hope when we have actually these observations, these traces here, that we can infer what these distributions here are. Not just the individual processes, but how these parameters or these different cells behave differently with population, and how these parameters are related to each other. That's then, ideally you can map this to biological processes. That's the entire idea.
If you just look at steady state data, the last two look sort of similar, but the first one, I mean, what would happen with steady state data? Could you still identify the distribution from steady state data, for that example?
If you have the distribution here over the trajectories, you see that this is the average cell. This is the mean. You see that variance here. So
It seems to be a higher variance, yeah. Can you compute it specifically? Well, it doesn't matter.
I'll
Oh,
tell
great.
you how to.
Oh, great.
How we tried.
Okay, great.
So I'm going to talk about two different aspects here. So first of all, given some data, how we infer these characteristics of a mechanistic model from these observations. And the proposal here is to use so-called non-dynamics effects models. I'll show you structure in a minute. And the second task then is, once you have these parameter distributions, how do you infer what the relevant biological processes are that cause the observed variability? Because you may have source of variability somewhere in a network that propagates, and the question is, what are the sources? We're going to do this with one aspect, one approach of global sensitivity. Now for the concept for these nonlinear mixed effects. So what we are assuming is that we have a dynamic model for each individual cell line, and we assume that these cells have deterministic dynamics. The dynamics of the cells are identical. But what causes these differences between cells is different instantiations of the parameters of these models, and also potentially different initial conditions. So this is on the parameters for each individual cell line. Then we assume a measurement model, and especially when you have single cell traces, then the measurement uncertainty is usually not known, so we have to infer this as well. So this is the so-called first level here. So this is the level of the individuals in the population. Now we tie them together in a second level, and that's why they're called nonlinear mixed effects models, by the relation between an individual and the population. So the individual cell parameters are assumed to be related to each other through the distribution. So to make it a little bit more formal, so this first level, essentially, this is just a standard ODE system. What the states here, xi means state vector for cell I. This depends on the state, potentially inputs, and the alpha I are the kinetic parameters in the population. And each cell individually can also have different initial conditions. So the parameters for each individual cell are then just the kinetic parameters and the initial conditions, and we denote this by beta I, which is the vector for each individual cell. For the measurement models, these are the observations, where we assume that there is some function of the states. Then we have additive noise, and this noise can also depend on this current state of the system. So these are two parameters for this noise. Eventually, we need to estimate the assumption here is that we have Gaussian noise.
So what is alpha I?
Alpha I are the kinetic parameters for cell. It's the parameter vector for cell. So that's the first stage, at the level of individual cells. And then we have the relation between the individual and the population. And the assumption here is that there's a so-called population average, beta. It's in the statistical literature called the fixed effect. And then each individual realization for each individual cell is given by this average, by the individual random effects. They are distributed according to a distribution with mean zero and a covariance matrix.
This is just a hierarchical model.
It's a hierarchy.
Sorry, I'm confused. So you have beta I twice. Beta I is the kinetic parameters of the initial conditions for each cell, but then you also have beta I being the...
This is how the beta i's are distributed.
Okay.
And that's the realization for so the problem with this type of models, they're not new. They're used in pharmacokinetics and pharmacodynamics for a long time, but usually they employ very small models, so 1 or 2 states, up to 5 parameters. So it's a quite difficult estimation problem, primarily because this here scales sporadically with the number of parameters. And current methods can do this up to roughly 40 effective parameters, and effective parameters is just each of these entries here.
Sorry, so you're making this assumption of Gaussian noise
Should it be log normal?
in the parameters? Well, yeah, that's what I'm trying to figure out. I mean, some people use log normal, other people, I don't even believe either makes sense, but...
Okay, so the only strict assumption is that we have Gaussian measurement noise. This is just one instance. It could be another distribution that you can characterize essentially by the mean and the
And
We
even
don't make an explicit
I see.
example.
And even for the Gaussian measurement noise, it's really Gaussian because you're allowing this G function, which could distort the.
Yes,
So it's
it
really
can
not-
be distorted, but this is absolutely right. Assume that it's just to hear a little bit first about how this can be estimated. That's from the literature. It's essentially, again, yeast cells in the microfluidic environment. They get osmotic shocks externally, and the way how this particular string is constructed, this is the input, and then just the standard model of gene expression. So there's a sensor essentially here, and then you have a standard model of gene expression. This is relatively small model, so it has roughly 20 parameters that need to be estimated. And there are different ways how you can do this. So one approach that's called the naive approach is the following. First you take each individual cell, you get a point estimate for the parameters of each individual cell, and then just take the average and the variance of these estimates to construct the population distribution. So that's this NTS here. On the left you see the experimental data over time. This is the input here, this is the protein concentration.
So yes, this is the y-axis is the protein fluorescence.
It's fluorescence.
To normalize by cell volume.
It is, in this case, it's normalized by cell volume, yeah, it's a concentration. So the data, the median are the black lines. These are the 95% quantities of the data in shading. And if you just look at the green curve here, then this approach gets the median pretty well, but the prediction error for the variances is completely. So that's the output here of this method here, this for one parameter distribution in the population over the parameters of the individual cells. So the other existing approach is called stochastic approximation expectation maximization. Is the algorithm in Randomness. We used it. So the basic idea is that you jointly estimate everything in this inference problem, not in these two stages, doing first the cells individually and then the population. And the output here is in blue. You see that again the median is well characterized, but also the variances as well. So the problem here is that this is an extremely computational intensive algorithm, and it's also pretty complicated, not very robust. So what we propose is an alternative for that estimation. So we call it global two-stage approach. Basic idea is the first is the following. So we have the first stage, we look at cells individually. So these are trajectories for individual cells. We fit our model to these trajectories just using standard solvers. And from this we get estimates for the parameters for each individual cell. And then we pour all these estimates. So in the second stage, each of these points here is an estimate for an individual cell. And what is now different from this naive approach is that in addition to taking into account what these point estimates are, we also take the uncertainty of these point estimates into account. And then try to come up with a joint estimate for the populations, this is here, where the influence of parameters for cells, like this green one here, that are outliers, perhaps just due to that you cannot really estimate it, they're weighed down and the others ones are over. Yeah, so that's the basic idea. We estimate the distribution here by taking the uncertainty.
You go back to the first stage of that. So it's not fully
we
basic.
we don't go back to the first stage. You could
You
do
can
it.
shrink.
We just to give you an idea how this actually works. So we have this covariance CI here is essentially the Fisher information matrix for each cell. So it characterizes the uncertainty in the individual estimates. What we're looking for are the beta and the D, are the mean and the covariance matrix of the population. We can write down the log likelihood, and then we take an EM type of algorithm. So first, the expectation step, we refine the estimates for the individual cell-specific parameters, taking both this uncertainty and the population level covariance into account. Then we do the M step, maximization step, for the population average, and then also for the population confidence. Then we iterate to this. It's the basic idea of the algorithm. And for the small example, if you now look at the results, so that's this GTS results, and the performance with respect to the mean, and also with respect to the variance in the small example, this is really comparable to the SAM. You also see that these distributions and they don't differ too wide. But now, what we did looked,
Could you compare to the true posterior in some examples?
we don't know what the true, we did make simulation studies to be able to compare to the true posterior. This is with real data, so we don't know the true posterior.
I mean, yeah, simulations as well.
Yeah, that's one. We did this controls. Just to give you an idea of scaling, so for the computational complexity, it scales with the number of cells. SAM really explodes pretty soon. The scale here is more benign. And what was interesting is now asking questions such as how much data do we actually need to do this inference for this particular problem? So we initially had roughly 500 cells. This Jakai index essentially quantifies the overlay, the confidence intervals that are predicted, and the data confidence intervals. And you see, even if you go down to roughly 100 cells, you get pretty good estimates. And it's also pretty insensitive to measurement noise. So for instance, here, it's fourfold the measurement noise just in simulations compared to the real measurement noise. It's still very good, and it drops. So it seems to be quite robust. Now I want to show you briefly the results from a more complicated study, a more complicated application. So the experimental setting is the following. So we have yeast cells here, and the green fluorescence comes from an amino acid transporter in the membrane of the yeast cells. And this particular one that has very high affinity for a particular amino acid, methyl. So in reality, in biology, what happens if you add methane into the medium, this transporter is no longer used, it's not very efficient. So it's being endocytosis and reduction degraded. So we add methane in here at time 9, and you see from the images that the fluorescence disappears. We can quantify this. So for instance here, the intensity of the cytoplasm of this particular transporter. And at time 0, it's relatively high, and then you see the individual trajectories on this. And there we ask the question, okay, what are the molecular causes for these observed phenotypic heterogeneity? So the model was pretty simple, so it's more pharmacological, but for instance here, this protein MOB1 needs to be transported to the cell membrane in order to be able to operate as a transporter. And this can be internalized to endosomes, can be recycled to the membrane here, can be degraded, and of course the protein is used as well. And this is externally controlled by the input, the amino acid, which has two effects. It inhibits the production, and it enhances the degradation by endosome. So this model is still relatively small for initial conditions for 40 parameters, but if you do this type of model, NLME, then effectively you have to run an estimate roughly 200 parameters.
How do you get to 200?
Sorry?
How do you get from 4 to 14 to 100? Like that number?
It's the covariance matrix parameter. It's 14 squared. And you have initial conditions are estimated and the measurement over. So this is now the comparison between model predictions and experimental data for different observables such as the cell volume, how much volume has the membrane, the intensity of the membrane, etc. The bottom line here is both with the GTS-OI method and in this case also with the SAM, we get pretty good predictions. They're consistent. So the problem then is how to integrate actually these estimates. Just to give you an idea, these are the probabilities, the estimated probabilities you can use over the parameters and conditions. And this is the correlation between these estimates that you can derive from this estimated D matrix, the covariance matrix. It's really hard to figure out what the structure of this is and what this means in terms of biological processes. So what it proposes is essentially trying to map the observed variability to variability in individual processes by variance decomposition. So what we do here is we look at the variance with respect to one of the parameters, J here, and this is the expectation with respect to all parameters function. So we do this ratio here, and these are the so-called first-order so-called indices. And just to show you briefly what the outcome is, or what the expected outcome is, how to interpret this. Again, to this small phi example here, we have two parameters, the degradation constant and the catalytic rate, so production constant. In this particular example, we have variability in this direction, which is the production constant here. And you see from the so-called indices, they are bound to 0 at 1. This variance in this kx in production fully explains the dynamics. Here, it can depend on time, so here we have the case where these are normally distributed. Initially, the production is involved in degradation, it takes some dynamics at later time points. We have the case where we cannot distinguish them, where they are correlated. So you see this by both being 1, so the sum of the larger. And now back briefly to our example. So that's essentially the result. It's a bit complicated, what it shows here. So these are parameters and initial conditions here. They're ordered, clustered according to absolute variance. These are the squares here. In each of these panels, over time, you have these so-called indices. And whenever they are colored, then they're significantly different from zero. Just to show you one or two highlights here. So for instance, here, these are observables related to growth, cell growing. And these are parameters related to initial conditions related to growth, and they have influence on these observables, but not on the intensities. The other interesting things here is, for instance, for this particular observation, how much protein is in the membrane. You see that this is time dependent, so initially the initial conditions are important, but then essentially this pathway here is important, not the maximum capacity, but the affinity of this pathway. And the other thing that you see here is that essentially that the absolute variance here is relatively small, but it explains a lot of the total variance in this observation. There are other things together. Bottom line here is if you try to decompose these variances, I think you can get interesting insights into which molecular processes determine which aspect of observed heterogeneity you can identify for researchers. That's a bit of a summary. So the proposal is to use this nonlinear mixed effects models. They make different approximations compared to stochastic analysis, because we assume that the dynamics of the variability comes from distributions of parameters. With the typical data quality and quantity you can now get from live cell imaging, actually you can do these types of estimations. The correlations are pose difficulties, also for interpretation. But the hope here is that we can use this type of framework in order to really identify which sub-processes in a larger network actually Just one final slide. So there's a number of really interesting open questions. So for instance, here Heather talked about identifiability yesterday. just to question how to disentangle these correlations in these estimates that result from poor identifiability versus ones that are real. Is an open question. These cells are also not independent. So for instance, this is the mother cell of this particular cell. So they're correlated across the lineage. And the question is how to incorporate this systematically into the models. And the final question that also relates to questions of, for instance, development. If you observe subpopulations of cells, it's neither necessary nor sufficient that you have, for instance, bimodal distributions in these parameters. So the question is how can we deal with that for what the actual causes are. So the most of the work was done by Lection Demaragem. She's currently looking for a postdoc position, and two other people in the group to this.
So, I mean, the idea of looking at your parameters to then work back and see what, where the noise is coming from in the final data, how robust, because you presuppose a certain structure of your model, how robust is that in terms of, you're trying to identify where it's come from, how robust is that to a different model structure? Like, let's say it was slightly different, then couldn't the answer be completely different?
Could be. So it's conditional on the model structure. We didn't try to do model selection. Another interesting.
Yeah, I was a little confused about the last point you made about the difference between bistability and the bimodal distribution. What do you mean by that? Because your initial state was part of the estimate, so the distribution initial state will be sort of capturing. I'm missing something.
And so the question is the following: so if you like in the, for instance, during stem cell differentiation, etc., eventually the cells form two different subpopulations. The question is whether it's say they were initially different, so they have, say, a bimodal distribution in the initial conditions or in the parameters, or if you simply have, say, a unimodal distribution in those, but then a process that leads to bifurcations, which you have a bistable element in between that drives this.
But wouldn't that be captured by the parameters or the initial conditions, the fact that you have this bifurcation, which are things that are estimated, so they should be part of your prior, or your, sorry. Well, we can talk offline.
Yeah, the problem with the current method is that we assume that we have a unimodal distribution. So
oh, I see. That's what you mean. I see. Okay, got it. But yeah.
So a very fundamental question. I think the standard gene expression model is initially proposed for like non-growing cells. And I think now you want to compare this standard gene expression with growing body yeast cells. So I guess you to compare the data meaningfully, you have to convert number to concentration to compare with the standard gene expression model. But then, I mean, you have to at some point you have to times the concentration with the volume constant.
So in this particular model I showed you in the end, that we have essentially the growth, we have volume as a dynamic state. So it starts to grow. Because it's a humanistic model, we were talking about concentrations. I didn't compare it to the standard stochastic model, just saying the structure of the reactions is this.
Yeah, but you approximate the dilution effect by degradation, in the model.
It's in the model, yes.
Yeah, but then you have to times some cell volume constant to compare with the model. Otherwise, the dimension is incorrect. You cannot use concentration to compare with the model.
And you compare with what? With the standard gene expression?
Whatever model, if you approximate cell volume growth by a degradation, you always have this problem. Like you want to use number to compare with the model, not concentration. Otherwise, the dimension is incorrect.
And so what we have, what we observe are these fluorescent signals. They are somehow related to their proportional to concentration. We don't actually this part of the measurement not to match between the observations, so the fluorescence and the media concentration. That's what is in the model.
But don't you have side scatter? You have scatter measurements, so you do normalize probably by volume to the concentrations in the pharmacometry data.
No, it's single cell microscopy.
Oh, single cell microscopy. Sorry. I was thinking of.
We have the volume from the images.
Yeah. I'm sorry. Okay, you'll.
Right, so you emphasize very clearly that this is based on deterministic modeling. And one obvious question, I guess, is how much sort of the noise in the reactions would affect the outcome. So if you would go back and
how do you propose to go back? So if I gave you a stochastic model of this dimension that essentially has 20 parameters and you want to estimate these parameters for a stochastic model, how would you do that?
well, so now you have parameters, let's say, and if you have good ideas about what the typical concentrations are, you have an idea about copy numbers, and in principle you could run a stochastic simulation and then see how that model then would behave and how similar it would.
Yeah, we did it in this case. It behaved quite similarly. Also, similar two things. First of all, we were rather in the high copy number regime for this purpose. One, and also the time scales are relatively short, so the gene expression dynamic stochastic fluctuations. We don't expect them to have such a big influence on this observation.
Is there actually a trade-off between how long you want to make the trajectories? Is there sort of an optimum in the length of these trajectories? Right? You mentioned relatively short trajectories, so this just triggered my mind. Is there actually an optimum in how long you want these trajectories to be?
I don't know.
I mean, longer it takes more CPU time, but maybe your estimate also becomes better.
So what we saw essentially here from this toy example, or the first example, is that the surprisingly little influence of the quality of the data on the estimates, on the estimation quality. So here, for example, that's a scenario where you just randomize the tracking, so you concatenate the trajectory part of the trajectory for one cell with another cell, just randomize this. And if you do this for whatever 90% of the cells, it's still pretty robust.